In this video, we are going to show you the general methods that can be used to harness microbes from environmental samples. So here we have compost, pommy and textile wastewater samples. For solid samples like compost, you can suspend the samples in distilled water. Here we suspend 2 grams of compost in 20 milliliters of distilled water that gives us 2% of initial sample. It is also important to do heat shock pre-treatment when using solid samples by placing the samples in 80 degrees Celsius oven. In this experiment, we are going to do pre-treatment for both compost and pommy samples. After the pre-treatment step, you can use the samples for zero dilution. However, if you are using a liquid sample like textile wastewater, the sample can directly be used for zero dilution. So here we are showing you how the zero dilution can be conducted using a textile wastewater. Make sure to apply a aseptic technique while doing this step. First of all, transfer 1 milliliter of sample into a tube consisting of 9 milliliters of distilled water. Then mix the solution properly. After that, transfer 1 milliliter of sample from the first tube into the second tube consisting of 9 milliliters of distilled water and mix the solution properly. Continue this serial step until the last remaining tube of distilled water. From the zero dilution, we now have diluted samples with the respective dilution factors as showing in this video. Next, we're going to transfer the diluted samples onto agar plates. The plates are labeled at the bottom. To further dilute our samples, we transfer 100 microliters of each diluted sample onto a plate. As you can see in this video, we start pipetting from the least concentrated sample to the most concentrated sample. By doing so, we can just use the same pipette tip for taking out the samples. Then flame a glass spreader and let it cool down for a few seconds before evenly spread the samples on the plates. Again, we start spreading the least concentrated sample to the most concentrated sample so that we don't have to reflame the spreader for each plate.
Okay.
The plates are placed in a 37 degrees Celsius oven for 24 hours. And these are the results.
Okay.
The controls are empty plates without samples, whereas the 10 to the power of minus 0 plate was made by directly transferring the samples without making dilution. The colonies obtained in these plates can be used for further physiological characterization. For an example, the bacterial growth profile, which could help to identify when is the best condition to start a treatment process using bacteria. Here we will be showing you how to do cell dry weight analysis and optical density analysis for the bacterial growth profiling. There are two methods for cell dry weight analysis, the centrifugation method and filtration method. For the centrifugation method, first we must record the width of the empty microcentrifuge tubes that will be used in the experiment. For filtration method, place a filter membrane in an aluminium boat as shown here and record the weight. Repeat this step for all filter membranes that will be used in the experiment. For optical density analysis, take out 1 milliliter of sample to check the optical density. Make sure to have triplicate for all your samples. It is recommended to check the optical density before continue with the dry cell weight analysis. Pengen lihat lagi. To continue with the centrifugation method, transfer 1 milliliter of sample into microcentrifuge tube. We recommend everyone to prepare your samples in triplicate. And then centrifuge the tubes in order to obtain the cell palette. Then wash the cell palette using distilled water. Repeat the washing step for 3 times for each sample. And on the last step, remove the supernatant and keep the cell palettes. Whereas for the filtration method, around 5 milliliters of sample is needed for each filtration. And remember to have triplicate for all your samples. Then place the membrane filter that have been weighed on the filter system and carefully assemble the system and turn on the vacuum pump. Next, wash the filter membrane with distilled water. Then pour the 50 milliliter sample onto the filter membrane. Wash the filter membrane for 3 times with distilled water. When that is done, place the filter membrane back into the aluminum boat. Make sure to place it in the same boat it was taken. Then place the cell palette tubes in the cell palette boat. Make sure to place the cell palette tubes in the same boat it was taken. Then place the cell palette tubes in the same boat it was taken. Then place the cell palette tubes in the same boat it was taken. Then place the cell palette tubes in the same boat it was taken. Then place the cell palette tubes in the same boat it was taken. Then place the cell palette tubes in the same boat it was taken. Then place the cell palette tubes in the same boat it was taken. Then place the cell palette tubes in the same boat it was taken. Then place the cell palette tubes in the same boat it was taken. Then place the cell palette tubes in the same boat it was taken. Then place the cell palette tubes in the same boat it was taken. Then place the cell palette tubes in the same boat it was taken. Then place the cell palette tubes in the same boat it was taken. Then place the cell palette tubes in the same boat it was taken. Then place the cell palette tubes in the same boat it was taken. Then place the cell palette tubes in the same boat it was taken. and filter membranes in oven until the weight is consistent, and record the final weight and proceed with data analysis.